So, dear friends, once again it's very hearty and warm welcome in this new lecture. And in this today's lecture, we are going to study in very detail the entire mechanism of fertilization in human being, and it is followed by the significance or importance of fertilization. Now let us start the study of this new topic, that is fertilization. So now let us start the study of this topic, that is fertilization. Actually, all of you are very well aware about the meaning of this term fertilization. We know that fertilization is considered as very important, very significant event in the process of sexual reproduction. We know that fertilization is nothing but it is the union or fusion of haploid male gamete, that is spermatozoon, and haploid female gamete, that is ovum, to form diploid zygote. So the meaning and definition of fertilization we are aware. Fertilization, it is the process of union or fusion of haploid male gamete, that is spermatozoon, and haploid female gamete, that is ovum, to form diploid zygote. Now in human being, not in human being, but in all mammals, the process of fertilization is internal. In human being, as in other mammals also, the process of fertilization is internal. It means the process of fertilization takes place in the body of female. Process of fertilization takes place in the body of female, and generally, usually, the fertilization takes place in the ampulla part of oviduct. We all are aware about the female reproductive system. You know the ampulla, which is middle, dilated and large part of the oviducts, or we can say fallopian tube. That ampulla is considered as the site of fertilization. It means in human being the fertilization is of internal type and it takes place in the ampulla part of fallopian tube, in the ampulla part of oviduct. Now here basically we people have to study about the different physical and chemical events taking place during the process of fertilization. खरं वास्तविक या टॉपिक मध्ये आपल्याला फर्टिलायझेशन ही प्रोसेस कशी घडून येते हे व्यवस्थित डिटेल समजून घ्यायचं आहे. That is the actual mechanism of fertilization, actual physical and chemical changes taking place during fertilization. But before to that, we are going to study about certain pre-fertilization events, about the different pre-fertilization events. Okay. So now let us talk about certain pre-fertilization events. Pre-fertilization events in the sense the different events taking place before the actual process of fertilization, before the actual union of male gamete and female gamete. Now the pre-fertilization events include three main activities. There are three main events. The first one is arrival of sperms. Arrival of sperms or arrival of spermatozoa, that is male gamete. I am going to talk with you. Second step or second event is arrival of ovum. Arrival of ovum, that is female gamete. And third step is capacitation of sperms or spermatozoa. Now to understand all these events taking place actually before fertilization, प्रत्यक्ष fertilization होण्याच्या आधी प्रत्यक्ष स्पर्मॅटोझोन आणि ओव्हम हे फ्यूज होण्याच्या आधी ज्या काही गोष्टी घडतात त्यांना या ठिकाणी आपण म्हटलंय प्री फर्टिलायझेशन इव्हेंट्स, आणि त्याच्यामध्ये या ठिकाणी तीन प्रमुख इव्हेंट्स आपल्याला कन्सिडर करायचे आहेत: अरायवल ऑफ स्पर्म्स, अरायवल ऑफ ओव्हम अँड कॅपॅसिटेशन ऑफ स्पर्म. हियर आय एम गोइंग टू ड्रॉ अ आउटलाइन डायग्राम ऑफ female reproductive system for our proper understanding. Look at the diagram. Here is supposed to be, you know, the uterus. All of you are known. Garbhashay. The main part of the uterus we all are known as the body, middle part, body. Then here is a cervix, the posterior or lower slightly narrow part, cervix, and here is the vagina. Here is the vagina. Here I am going to show. Here is the vagina. We know that vagina opens outside by means of vaginal opening. Also we know here are presence of oviducts, that is fallopian tube. These are paired structure, but I am going to show here temporarily single structure. So here I have shown only single ovary, single oviduct. Here is a uterus, that is the body of uterus. Here is cervix and here is the vagina. Now we have to study first pre-fertilization event, that is arrival of sperms, arrival of male gametes, that is sperms or spermatozoa. During sexual intercourse, that is during mating of male and female, that is during coitus, you know, coitus, coitus. This term is used for the sexual mating of male and female, for the sexual intercourse. So during coitus, during sexual intercourse, during copulation, we can say the penis of male is entrusted into the vagina of female. The penis of male is entrusted into the vagina of female, and male discharges about 3 to 5 ml of semen, 3 to 5 ml of semen high up in the vagina, close to the cervix, high up in the vagina, close to the cervix. You know, this process is actually called as insemination. This act is called as the insemination. Insemination means deposition of semen from male into the female reproductive tract. All of you are known semen, that is male reproductive fluid, and during single ejaculate contain about 3 to 5 ml of semen, and this 3 to 5 ml of semen contain about 300 to 450 millions of sperms. Here that millions of sperms along with semen get deposited in the upper part of vagina close to the cervix. This act is called as the insemination. Now the sperms present here in the semen reach to the fallopian tube, reach to the ampulla part of fallopian tube, because we know the ampulla, it is the site of fertilization. Repeatedly I told you, हा fallopian tube चा किंवा oviduct चा हा जो मिडल वाइड पार्ट आहे ज्याला आपण ampulla असं म्हणतो, या ठिकाणी प्रत्यक्ष फर्टिलायझेशन घडून येतं, आणि म्हणून ऑब्वियसली ह्या स्पर्म्सला, सिमेन मध्ये असलेल्या स्पर्म्सला इथपर्यंत पोहोचावं लागेल. So that sperms have to reach up to the ampulla. Now the sperms reach up to the ampulla partly by taking its own efforts. याचा अर्थ अंशतः स्पर्म्स स्वतः प्रयत्न करतात त्या ठिकाणी एम्पुला पार्ट पर्यंत पोहोचण्यासाठी अँड दॅट स्पर्म्स आर फॅसिलिटेटेड बाय सर्टन ऍक्टिव्हिटीज ऑफ फिमेल रिप्रोडक्टिव्ह ट्रॅक्ट। याचा अर्थ त्या स्पर्म्सला इथपासून या ठिकाणी एम्पुला पर्यंत पोहोचण्यासाठी ही फिमेल रिप्रोडक्टिव्ह ट्रॅक्ट जी आहे, फिमेल रिप्रोडक्टिव्ह सिस्टमचे जे भाग आहेत, दॅट इज वी कॅन सर्व्हिक्स, युटेरस, फॅलोपियन ट्यूब, दे आल्सो पार्टली सपोर्ट। सो व्हाट डिड आय से? I said that the sperms reach up to the ampulla part of fallopian tube partly by their own efforts and partly by certain activity of uterus, cervix and fallopian tube. The sperms we know are motile structure. The sperms are motile structure. They have ability of independent movement and that's why the sperms they travel across the length of uterus. They travel across the length of uterus by the lashing movement of their tail. It means the sperms swim in fluid medium. या ठिकाणी युटेरस मध्ये आपण मागे शिकलो की युटेराइन फ्लुइड सिक्रेट केलं जातं। युटेरसच्या एंडोमेट्रियल ग्लँड्स काहीतरी लिक्विड फ्लुइड सिक्रेट करतात, अर्थात या ठिकाणी असणारे जे फ्लुइड आहे त्या फ्लुइड मध्ये हे स्पर्म्स स्विमिंग करतात बाय द लॅशिंग मूवमेंट ऑफ देअर टेल, व्हीप लाइक मूवमेंट ऑफ देअर टेल, अँड द स्पर्म्स दे ट्राइज टू रीच अप टू द एम्पुला पार्ट बाय देअर ओन एफर्ट। स्वतःहून ते तिथपर्यंत पोहोचण्याचा प्रयत्न करतात। द स्पर्म्स स्विम at the rate of about 1.5 to 3 millimeter per minute. 1.5 to 3 millimeter per minute. अर्थात हे स्पर्म्स या ठिकाणाहून इथपर्यंत पोहोचण्यासाठी त्यांचा जो मोटिलिटीचा रेट आहे तो आता मी तुम्हाला सांगितला, दॅट इज 1.3 टू 1.5 टू 3 मिलीमीटर पर मिनिट। एका मिनिटाला 1.5 ते 3 मिलीमीटर एवढं अंतर ते स्पर्म्स क्रॉस करत असतात। दिस इज द एफर्ट टेकन बाय स्पर्म्स इटसेल्फ, बट also I told you the female reproductive tract also facilitate that sperm, also support that sperms to reach up to the ampulla. How is that? We have studied last time the semen contain prostaglandins. Semen contain prostaglandin. Do you remember? Semen is the mixture of secretion of seminal vesicle, prostate gland, and that contain prostaglandin, just a material like the hormones. That prostaglandin present in the semen, या semen मध्ये असणारो prostaglandin नावाचे जे पदार्थ आहेत, they induces, they stimulate the cervix, uterus and fallopian tube to undergo slight contraction, because of which the sperms are supported, facilitated to move upward. मी काय म्हटलं मराठीतून सांगतो, या सीमेन मध्ये जे काही प्रोस्टाग्लॅंडिन्स असतात त्या प्रोस्टाग्लॅंडिन्स मुळे सर्व्हिक्स, युटेरस आणि फॅलोपियन ट्यूबचं स्लाइट कॉन्ट्रॅक्शन होतं आणि त्या कॉन्ट्रॅक्शन मुळे सुद्धा स्पर्म्सला इथपासून फॅलोपियन ट्यूब पर्यंत पोहोचण्यासाठी मदत होत असते। एवढंच नाही, यू प्लीज लुक ॲट द डायग्राम. दिस सर्व्हिक्स इज एक्सटेंडेड इनटू द वजायना. सर्व्हिक्सचा भाग या ठिकाणी जणू काही वजायनामध्ये असा एक्सटेंड झालेला असतो, लोंबकळत असतो. अँड दिस सर्व्हिक्स इट शोज aspiration. It aspirates the sperm. Aspirates means what? It is a sort of suction. You know, whenever we drink any fruit juice, whenever we drink any cold drink, at that time we put the straw in that bottle of cold drink and we suck that cold drink. That is nothing but the aspiration, sucking. In the same manner, this cervix, it aspirates the sperms. So this aspiration, this aspirating action shown by the cervix and contraction of uterus and fallopian tube, it facilitates the sperms to reach up to the fallopian tube. Here we were discussing only about the arrival of sperms. Now I said here the semen contain millions of sperms. Semen contain millions of sperms, isn't it? But out of that millions of sperms, only few hundreds of sperms can reach up to the ampulla. 100 or so. 100, 150, 200, 300 एवढे स्पर्म्स या ठिकाणी फक्त एम्पुला पर्यंत पोहोचू शकतात. Why this is so? There are different reasons. Say, first of all, the acidic nature of vagina can kill the sperms. आपल्याला माहिती आहे या ठिकाणी वजायना ऍसिडिक असते, त्यामुळे काही स्पर्म्स त्या ठिकाणी नष्ट होऊन जातात. सेकंड, सम ऑफ द स्पर्म्स कॅन बी डाइड ड्यू टू द युटेराइन सिक्रीशन. त्या युटेरस मध्ये जे सिक्रीशन्स तयार होतात त्यामुळे काही स्पर्म्स नष्ट होऊन जातात. तिसरं, वी कॅन से ड्यू टू द लॉस ऑफ एनर्जी. Due to the loss of energy of that sperms, the sperms cannot reach up to the ampulla part. And one more, due to the engulfing phagocytes. Here in the uterine endometrium, uterusचा जो endometrium आहे त्याच्यामध्ये आपल्याला माहिती आहे that is vascular, highly vascular, having a rich supply of blood, and blood contains some phagocytic cells, you know WBCs, white blood cells, पांढऱ्या रक्तपेशी. काही पांढऱ्या रक्तपेशी या फॅगोसाइटिक असतात. फॅगोसाइटिक म्हणजे काय आपल्याला माहिती आहे, दे शो फॅगोसाइटोसिस, दॅट इज दे एंगल्फ अँड डायजेस्ट द डिफरंट फॉरेन मटेरियल. अर्थात या ठिकाणी फिमेल बॉडीच्या दृष्टीने स्पर्म्स हे फॉरेन बॉडीज आहेत आणि म्हणून या ठिकाणी त्या वॉन्डरिंग फॅगोसाइट्स अनेक स्पर्म्सला खाऊन टाकतात, नष्ट करत असतात. अँड दॅट्स व्हाय दो मिलियन्स ऑफ स्पर्म्स गेट डिपॉझिटेड हियर, only 100, 200, 300, few hundreds of the sperms can reach up to the ampulla part of oviduct. This is all about the arrival of sperms. Now come to the arrival of ovum. We know that ovum, that is female gamete, and the ovum is produced by the ovary. We have already studied the Graafian follicle. Graafian follicle, the largest mature well-grown follicle, it burst off, rupture, and it shed out the ovum. And this ovum shedded out from the ovary is not true ovum. It is not true ovum. It is the developmental stage of ovum, that is secondary oocyte, isn't it? Secondary oocyte. Last time we have studied again. Haploid secondary oocyte. And the secondary oocyte has arrested its next maturation division, second maturation division, in the metaphase second of meiosis second. You people have to remember all these things. Anyway, so this secondary oocyte released out from the ovary. Ovary मधून हे जे secondary oocyte shed out केलं जातं, we know that is captured and pushed into the infundibulum, and it is brought here in the ampulla part of oviduct by the peristaltic and ciliary action. अर्थात या ठिकाणी हे सेकंडरी oocyte इथे येऊन थांबलेलं असतं ampulla part मध्ये. This is the arrival of ovum. You must know that the ovum, which is in secondary oocyte stage, that can survive, that can live for about 72 hours after ovulation. याचा अर्थ जवळपास 72 तास ते या ठिकाणी अस्तित्वात असतं. बट इट कॅन बी फर्टिलाइज्ड विदिन 12 to 24 hours आफ्टर ओव्युलेशन. मात्र फर्टिलायझेशन केव्हा सक्सेसफुल होतं? फक्त ओव्युलेशन झाल्यानंतर पुढच्या 12 ते 24 तासामध्ये. Later on that secondary oocytes degenerates, and that is, we can say, again phagocytosed by the WBCs. Anyway, now the third pre-fertilization event is capacitation of sperms. Capacitation of sperms means what? In the female reproductive system, the sperms are made capable to fertilize the ovum. In the female reproductive system, the sperms are made capable to fertilize the ovum. याचा अर्थ या ठिकाणी स्पर्म्स फिमेल रिप्रोडक्टिव्ह सिस्टीम मध्ये आल्यानंतर खरं वास्तविक त्यांना ओव्हमला फर्टिलाइज करण्यासाठी एबल बनवलं जातं, तयार केलं जातं. हाऊ इज दॅट? the uterine secretion or we can say the secretions of female reproductive tract. या फिमेल रिप्रोडक्टिव्ह ट्रॅक्ट मध्ये तयार होणारे, युटेरस मध्ये तयार होणारे जे काही सिक्रिशन्स असतात, जे काही स्त्राव असतात, that some of the secretion they remove some molecules present on the head of sperm. Present on the head of sperm. You know, here is supposed to be the head of sperm. We have already studied in previous lecture, isn't it? Here is the head of sperm. अर्थात there are presence of some material, there are presence of some molecules on the surface of head of sperm, and they are removed by the secretions of female reproductive tract. And this act is called as the capacitation of sperms. Now here we have discussed about the pre-fertilization events. That is, after insemination, the sperms reaches up to the fallopian tube. After ovulation, the ovum has reached into the ampulla part, and now that sperms are capacitated, they have acquired the ability of fertilization. त्या स्पर्म्सला ओव्हमला फर्टिलाइज करण्याची क्षमता प्राप्त झाली आहे, आणि असे हे जे कपॅसिटेटेड स्पर्म्स आहेत, ज्यांना फर्टिलायझेशन करण्याची क्षमता प्राप्त झाली आहे, such capacitated sperms now they reach up to the ovum, and now it actually starts the process of fertilization. आता प्रत्यक्ष आपल्याला शिकायचं आहे the different physical and chemical events taking place during the process of fertilization. So let us talk about it. So now, after the discussion of pre-fertilization events, let us start the discussion and study of actual different physical and chemical events taking place during the process of fertilization. Actually, during fertilization, different events occur. We say, first of all, the penetration of sperm into the ovum. Penetration of sperm into the ovum is followed by the activation of ovum. What does it mean? We shall study. And it is followed by the amphimixis, formation of synkaryon, that is the actual formation of zygote. So we have to study these events definitely. But to understand all these events, here I am going to draw a proper diagram of ovum again. So please look at the diagram. We have already studied the ovum before fertilization is in secondary oocyte stage. Please look at the diagram. We have already studied that the ovum in secondary oocyte stage has a plasma membrane. You know, this plasma membrane is called as the oolemma or vitelline membrane. In our previous topic we have studied this is nothing but the plasma membrane of ovum. Also we have seen the cytoplasm. The cytoplasm of ovum is differentiated into peripheral, thick and granular part known as egg cortex. या ठिकाणी हा जो सायटोप्लाझम आहे त्याचा पेरिफेरल जो पार्ट आहे, पेरिफेरल रिजन मध्ये हा सायटोप्लाझम थोडा थिक असतो, विस्कस असतो आणि ग्रॅन्युलर असतो. ग्रॅन्युलर याचा अर्थ it shows presence of few granules. Actually these granules are nothing but the tiny vesicles. हे granules म्हणजे लहान लहान vesicles आहे, यांना आपण cortical granules म्हणतो असं मी सांगितलं होतं. Let me label. Here is supposed to be the cortical granules. These are cortical granules because these granules, these small particles, these tiny vesicles are present in this peripheral region of cytoplasm, which is thick, and you know it is called as the egg cortex. Then the inner part of the cytoplasm is known as ooplasm. Here is supposed to be the ooplasm. And in this ooplasm, nucleus is present eccentrically, slightly away from the center. And that nucleus is haploid. It contains 23 chromosomes. 23 chromosomes. I would like to add here one point, that chromosomes of the ovum are not organized into the nucleus. Chromosomes of the ovum are not organized into the nucleus. That 23 chromosomes are like this. These are not wrapped, these are not covered into the nuclear membrane. They are not organized into the nucleus. So, and here in the ooplasm, different cell organelles are present except centriole. Mind well, we have already discussed. In addition to that, we know the oolemma is surrounded by a non-cellular thin gelatinous layer. Non-cellular thin gelatinous layer. That is nothing but the zona pellucida. This is zona pellucida. It is non-cellular layer made up of gelatinous material, sticky material. Zona pellucida. There is a little space between zona pellucida and oolemma called as the perivitelline space. And we know here towards the animal pole, here is the presence of first polar body. First polar body we have already studied. Now again, around this zona pellucida there is a presence of the layer of tall column-like compactly arranged cells. Tall column-like compactly arranged cells. These cells togetherly form a layer just outside the zona pellucida, and you know this layer is called as the corona radiata. Corona radiata. अर्थात या ठिकाणी या corona radiata च्या cells आहेत. Here I have shown the corona radiata cells, and also may be presence of few other follicular cells. त्याच्या भोवती काही follicular cells पण असतात. काही follicular cells पण असतात. These are few follicular cells, folliculture cells. आता या ठिकाणी मी स्पर्म पण दाखवतोय आणि प्रत्यक्ष आपण फर्टिलायझेशन ची प्रोसेस या ठिकाणी सुरू करतोय. The first step is entry of sperm into the egg. Entry of sperm into the egg. Entry of sperm into the egg. I have told you, out of millions of sperms, only few hundreds of sperms can reach up to the ovum, and out of that few hundreds of sperms, only one sperm gets success. Only one spermatozoon gets success to penetrate the ovum, to fertilize the ovum. That is, generally the sperm penetrates the ovum from the nearer to the animal pole. Last time we have studied this ovum, this was polarity. The end towards which nucleus and first polar body is present is called as the animal pole. आपल्याला आठवत असेल की या सेकंडरी oocyteचं न्यूक्लियस आणि फर्स्ट पोलार बॉडी ज्या बाजूला आहे त्याला आपण एनिमल पोल म्हणतो, आणि जे अपोजिट एंड आहे त्याला आपण म्हणतो व्हेजिटल पोल. सर्वसाधारणपणे एनिमल पोल कडून, एनिमल पोल जवळून एंट्री करणारा, पेनिट्रेट करणारा जो स्पर्म्स असतो त्याला या ठिकाणी पेनिट्रेशन करण्यामध्ये सक्सेस मिळत असतं. नाउ हियर आय एम गोइंग टू शो द स्पर्म. Look, we know that the sperm has anterior part, that is nothing but the head. Head is followed by neck. Neck is followed by middle piece. All of you are known. Here is the middle piece and here is the tail. Also we have seen here the head. It encloses a single haploid nucleus with the, of course, with 23 chromosomes, 23 chromosomes, and the tip of the head is covered by a cap. What is that? Acrosome. Here is acrosome, isn't it? At the tip, anterior end of head of sperm, a cap-like structure is present called acrosome. Here is the neck. We know the neck, it contains two centrioles, proximal and distal centriole. From distal centriole, it starts the axial filament which runs from the middle piece, and also it is extended as a tail. That we all are know. Now, the capacitated sperm, when reaches the ovum, you know capacitated sperm, I told you few minutes before, the sperm which has acquired the ability to fertilize the ovum. ओव्हमला फर्टिलाइज करण्याची क्षमता प्राप्त झालेला जो स्पर्म त्याला आपण म्हणतो कपॅसिटेटेड स्पर्म. नाउ व्हेन दिस कपॅसिटेटेड स्पर्म reaches near the ovum, usually near the animal pole, the acrosome, look, this acrosome, this cap-like structure, this acrosome, it releases a few enzymes, namely hyaluronidase and corona penetrating enzyme. Hyaluronidase, this enzyme dissolves the follicular cells, these follicular cells, because here the ground substance or cementing substance of that follicular cells is hyaluronic acid. लक्षात घ्या, ह्या ज्या follicular cells आहेत त्या एकमेकांना ज्या सबस्टन्स ज्या मटेरियल मुळे चिकटून बसलेले आहेत ते आहे हायलुरॉनिक ऍसिड, आणि त्या हायलुरॉनिक ऍसिडला नष्ट करण्याचं काम हे एन्झाइम करतं, हायलुरॉनिडेज. पर्यायाने द follicular cells that get destroyed, and corona penetrating enzyme, it also dissolves the cells of corona radiata, and now this sperm, it moves forward and reaches near the zona pellucida. Let me show in the diagram. What did I say? When the capacitated sperm reaches near the ovum, generally towards the animal pole, near the animal pole, the acrosome of sperm, it releases enzymes hyaluronidase and corona penetrating enzyme. Due to this enzyme, the follicular cells also get dissolved, broken down. The cells of corona radiata are also dissolved, and the sperm, it now reaches near the zona pellucida, isn't it? Zona pellucida. Look at the diagram. या ठिकाणी आपण म्हणूया की आता हा जो स्पर्म आहे त्याचं हेड या ठिकाणी आलेलं आहे. Let me show properly. Now when the head of sperm reaches the zona pellucida, the acrosome, the cap-like structure, this acrosome again releases one more enzyme. आता या ठिकाणी हे acrosome परत एक एन्झाइम सिक्रेट करतं. त्या एन्झाइमचं नाव आहे झोनालायसिन. Or it may be called as acrosome. Zonalysine or acrosome. Name itself indicates zonalysine. Lysine means what? Which has ability to bring about lysis. Lysis means breaking down, to dissolve. या ठिकाणी हे जे एन्झाइम आहे झोनालायसिन ते कशाला तरी ब्रेक करणार आहे, कशाला तरी डिसॉल्व करणार आहे. कशाला डिसॉल्व करेल? झोना, this word stands for the zona pellucida. So the zonalysine, which is also called acrosine, it dissolves the zona pellucida. Zona pellucida हे dissolve होऊन जातं, and now the head of sperm, it come closer to the ulema. Look at the diagram. या ठिकाणी स्पर्मचं जे हेड आहे ते आता प्रत्यक्ष उलेमाला टच झालेलं आहे. उलेमाला टच झालं म्हणजे कोणाला? vitelline membrane, that is nothing but the plasma membrane of ovum, right? And when it touches the sperm, it occurs one very important reaction, fertilizing anti-fertilizing compatibility reaction. What does it mean? It means the zona pellucida has some glycoprotein molecules on it. कृपया लक्षात घ्या, हे जे झोना पेल्युसिडा आहे, ह्या झोना पेल्युसिडावर काही ग्लायकोप्रोटीनचे मॉलिक्युल्स असतात. तुम्हाला माहिती आहे ग्लायकोप्रोटीन इट इज अ कॉन्जुगेटेड प्रोटीन. या ठिकाणी 3 प्रकारचे ग्लायकोप्रोटीनचे मॉलिक्युल्स या ठिकाणी झोना पेल्युसिडावर असतात. त्यांना ZP1, ZP2, ZP3 असं म्हणतात. ZP stands for the zona pellucida. Zona pellucida glycoprotein first, zona pellucida glycoprotein second, zona pellucida glycoprotein third. असे 3 प्रकारचे ग्लायकोप्रोटीन मॉलिक्युल्स प्रेझेंट असतात. कुठे? ऑन द झोना पेल्युसिडावर. ऍक्च्युअली दॅट ग्लायकोप्रोटीन मॉलिक्युल्स acts as sperm receptor proteins. Sperm receptor proteins. नावच आपल्याला सांगतं. रिसेप्टर मीन्स टू रिसीव. याचा अर्थ स्पर्मला रिसीव करणारे प्रोटीन्स. मग या ठिकाणी मी असं म्हटलं की झोना पेल्युसिडा इट शोज 3 डिफरंट टाइप्स ऑफ ग्लायकोप्रोटीन मॉलिक्युल्स, आउट ऑफ विच ZP3 या प्रकारचा जो ग्लायकोप्रोटीन मॉलिक्यूल आहे इट ऍक्ट्स ॲज स्पर्म रिसेप्टर प्रोटीन. अँड दिस ZP3 is also known as fertilizing molecule. Fertilizing याला fertilizing molecules पण म्हणतात. In the same manner, on the head of sperm, त्याच पद्धतीने या ठिकाणी स्पर्मच्या हेडवर सुद्धा काही प्रोटीन मॉलिक्युल्स असतात. त्यांना anti-fertilizing molecules म्हणतात. Try to understand. Anti-fertilizing molecules. Anti-fertilizing molecules means these are protein molecules present on the surface of head of spermatozoon. लक्षात घ्या, या स्पर्मॅटोझोनचं जे हेड आहे त्याच्या सरफेसवर जे प्रोटीन मॉलिक्युल्स असतात त्यांना म्हटलं जातं अँटी फर्टिलायझिंग प्रोटीन मॉलिक्युल्स. आणि झोना पेल्युसिडावर जे ग्लायकोप्रोटीन्स मॉलिक्युल्स आहेत ज्यांना आपण ZP3 म्हटलं त्यांना म्हटलं जातं फर्टिलायझिंग मॉलिक्युल्स. ऍक्च्युअली दिस फर्टिलायझिंग अँड अँटी फर्टिलायझिंग दे शो कॉम्प्लिमेंटरी ऍक्शन. They show compatibility. It means matching or non-matching nature. It means only the matching or compatible anti-fertilizing can bind with the particular fertilizing. याचा अर्थ विशिष्ट प्रकारचे अँटी फर्टिलायझिंग मॉलिक्यूल विशिष्टच प्रकारच्या फर्टिलायझिंग मॉलिक्यूलला बाइंड होतील. याचा फायदा काय होईल? याचा फायदा असा होईल कि a sperm of particular species can fertilize the ovum of particular species. It is called fertilizing anti-fertilizing compatibility reaction. So I was telling you, after destruction of zona pellucida, the sperm head touches the oolemma. आता या ठिकाणी झोना पेल्युसिडाला डिसॉल्व करून हे स्पर्मचे हेड या ठिकाणी प्लाझ्मा मेम्ब्रेन पर्यंत आलंय, oolemma पर्यंत आलंय, व्हायटेलाइन मेम्ब्रेन पर्यंत आलेलं आहे. And now the plasma membrane of head of sperm and plasma membrane of ovum, they fuse together. Look at the diagram. They fuse together. Plasma membrane of head of sperm and plasma membrane of oolemma, vitelline membrane, plasma membrane of ovum, they fuse together. At the same time, this plasma membrane of ovum, it produces some finger-like projections around the sperm head. या ठिकाणी हा जो प्लाझ्मा मेम्ब्रेन आहे ओव्हमचा, किंवा आपण ज्याला उलेमा असं म्हणतो, तो या ठिकाणी या स्पर्मच्या हेड भोवती अशा काही फिंगर लाईक प्रोजेक्शन्स तयार करतो. त्यांना मायक्रोविलाय म्हणतात. अँड दिस मायक्रोविलाय constitute fertilization cone. These microvilli togetherly known as the fertilization cone. याला म्हटलं जातं fertilization cone. Means fertilization cone is nothing but the microvilli, small finger-like structures formed by the vitelline membrane around the sperm head. Means actually it welcomes the sperm head to enter into the ovum. So this structure is called as the fertilization cone. Here we have studied the first step in the process of fertilization, that is entry of sperm into the egg. Now what happens? Second step, activation of ovum. Let me explain you what is second, activation of ovum. Once again, please look at the diagram. I said just now, as soon as the plasma membrane of head of sperm and plasma membrane of ovum fuse with each other, it stimulates, listen, it stimulates the secondary oocyte, isn't it? Yet it is secondary oocyte, and it has arrested in the meiosis second maturation division. तुम्हा लोकांना आठवतं का बघा, आपण पाहिले की हे सेकंडरी oocyte त्याचं सेकंड मॅच्युरेशन डिव्हिजन सुरू करतं, पण ते सेकंड मॅच्युरेशन डिव्हिजन हे थांबवलेलं असतं, स्टॉप केलेलं असतं, अरेस्ट केलेलं असतं. कोणत्या स्टेजला? मेटाफेज सेकंड. मग या ठिकाणी ते जे सेल डिव्हिजन आहे meiosis second maturation division, that resumes, ते पुन्हा सुरू होतं. And this secondary oocyte completes that secondary maturation division, meiosis second, and it leads to the formation of two highly unequal cells. म्हणजे या सेकंडरी oocyteचं सेकंड मॅच्युरेशन डिव्हिजन पूर्ण होतं आणि त्याच्यापासून पुन्हा दोन अनइक्वल सेल्स तयार होतात. One is very large and another is very small. You know, this large cell is nothing but the true ovum, and that small cell is nothing but the second polar body. तुमच्या लक्षात असावं, second polar body. याचा अर्थ इथे second polar body जी तयार होते ही काय रिप्रेझेंट करते? This second polar body represent the entry of sperm into the ovum. It means now here actual true ovum is formed. You know, ovum is formed means process of oogenesis get completed. याचा अर्थ खऱ्या अर्थाने आता oogenesis पूर्ण झालं. आपण मागच्या टॉपिक मध्ये एकदा शिकलो. मी तुम्हाला oogenesis शिकवताना म्हटलं, the process of oogenesis it begins before the birth of female baby and completes only during fertilization, that is the penetration of sperm into the ovum. म्हणजे या ठिकाणी ऍक्टिव्हेशन ऑफ ओव्हम घडून येत असताना पहिला बदल काय होतो, तर हे सेकंडरी oocyteचं सेकंड मॅच्युरेशन डिव्हिजन होतं आणि त्या ठिकाणी ट्रू ओव्हम तयार होतं आणि एका बाजूला सेकंड पोलार बॉडी तयार होते. दुसरा बदल अत्यंत महत्त्वाचा, as soon as the sperm penetrates the ovum, these cortical granules, look, cortical granules, isn't it? These cortical granules, they stuck to the vitelline membrane. हे cortical granules या ठिकाणी पेरिफेरीला ढकलले जातात आणि ते या ठिकाणी या उलेमाला बाइंड होतात, अशा प्रकारे. They bind to the ulema. And these cortical granules, they secrete some enzyme into the perivitelline space. हे कॉर्टिकल ग्रॅन्युल्स या पेरिव्हायटेलाइन स्पेस मध्ये या गॅप मध्ये काही एन्झाइम सिक्रेट करतात, ड्यू टू विच द झोना पेल्युसिडा becomes quite tough, hard. It is necessary to prevent the further entry of sperm. It prevents the entry of another sperm into the ovum. म्हणजे या ठिकाणी हे झोना पेल्युसिडा हार्ड झाल्यामुळे फर्टिलायझेशन मेम्ब्रेन पण असं त्याला म्हणतात. हे तयार झाल्यामुळे आता पुन्हा नवीन स्पर्म ओव्हम मध्ये एंट्री करू शकणार नाही. इट मीन्स इट हेल्प्स टू प्रिव्हेंट पॉलिस्पर्मी अँड मेंटेन मोनोस्पर्मी. प्रिव्हेंट पॉलिस्पर्मी याचा अर्थ काय? एंट्री ऑफ मोर स्पर्म इनटू द ओव्हम. या ठिकाणी झोना पेल्युसिडा थिक आणि टफ झाल्यामुळे त्या ठिकाणी पुन्हा नवीन स्पर्म या ठिकाणी ओव्हम मध्ये प्रवेश करू शकत नाही, एंट्री करू शकत नाही, म्हणजे पॉलिस्पर्मी शक्य नाही आणि एकच स्पर्म आतमध्ये प्रवेश करायला त्या ठिकाणी परमिट केलं गेलंय, अर्थात मोनोस्पर्मी इज मेंटेन. It means during the activation of ovum, two major changes occur. First, the second maturation division of secondary oocyte is completed, formation of ovum, and दूसरा here is the vitelline membrane is converted into fertilization membrane necessary to maintain the monospermy. Now come to the third and final step, that is nothing but the amphimixis. Or karyogamy. It may be called as the syngamy, but most proper term karyogamy or amphimixis. Amphi याचा अर्थ बोल, mixis याचा अर्थ mixing, आणि karyo stands for nucleus, gamy stands for here fusion of nuclei. Now look, during sperm penetration, now the nucleus of sperm enters into the ovum. Nucleus of sperm enter into the ovum. या ठिकाणी जो न्यूक्लियस आहे, हॅप्लॉइड न्यूक्लियस स्पर्मचा, तो या ठिकाणी ओव्हम मध्ये ढकलला जातो. It is also followed by the proximal centriole. त्याच्या पाठोपाठ नेक मध्ये असणारा एक जो सेंट्रिओल आहे, आपल्याला माहिती आहे नेक मध्ये दोन सेंट्रिओल्स असतात, प्रॉक्सिमल आणि डिस्टल. त्यापैकी प्रॉक्सिमल सेंट्रिओल पण ओव्हम मध्ये येतो. मीन्स देयर इज अन इंट्रोडक्शन ऑफ सेंट्रिओल इनटू द ओव्हम व्हिच इज इनिशियली ॲब्सेंट इन मॅच्युअर ओव्हम. जो ओव्हम मध्ये सुरुवातीला सेंट्रिओल नसतो, असं मी अनेक वेळा सांगितलं, तो सेंट्रिओल या ठिकाणी आता स्पर्मच्या माध्यमातून ओव्हम मध्ये टाकला जातो. अँड रिमेनिंग पार्ट, दॅट इज मिडल पीस टेल, इज डिस्कार्डेड आउट. बाकीचा भाग मात्र बाहेर राहतो. या ठिकाणी अर्थात स्पर्मची एंट्री झाल्यानंतर इथे परत आपण म्हणूया की फर्टिलायझेशन मेम्ब्रेन तयार होतं अँड फर्दर एंट्री ऑफ स्पर्म इज नॉट पॉसिबल. नाउ व्हॉट हॅपन्स? Now this nucleus of sperm, listen, this nucleus of sperm is called as male pronucleus. Male pronucleus. The nucleus of sperm is called male pronucleus. Pronucleus याचा अर्थ the nucleus of sperm before fertilization. प्रत्यक्ष फर्टिलायझेशन होण्याच्या आधी त्याला म्हटलं जातं मेल प्रोन्यूक्लियस. आणि या ठिकाणी मी सांगितलं दीज आर द क्रोमोझोम्स ऑफ ओव्हम. 23 क्रोमोझोम्स ओव्हमचे, दे आर नॉट ऑर्गनाइज्ड इनटू द न्यूक्लियस, बट देन अँड देन दीज 23 क्रोमोझोम्स ऑफ द ओव्हम इज नोन ॲज फिमेल प्रोन्यूक्लियस. So here the male pronucleus contain 23 chromosomes, female pronucleus contain 23 chromosomes. Now the nuclear membrane of the male pronucleus disintegrates, and thus that 23 chromosomes of the male pronucleus becomes free. या ठिकाणी मेल प्रोन्यूक्लियस चे 23 गुणसूत्र फ्री झाले, या ठिकाणी फिमेल प्रोन्यूक्लियस चे 23 गुणसूत्र फ्री झाले. नाउ दिस सेंट्रिओल, हा जो सेंट्रिओल आहे, that centriole also divides into two daughter centrioles. या सेंट्रिओल पासून दोन डॉटर सेंट्रिओल्स तयार होतात, and these daughter centrioles they form spindle. त्यांच्यामध्ये स्पिंडल तयार होतं. स्पिंडल तयार होतं आणि हे स्पिंडल इट हेल्प्स फॉर द मिक्सिंग ऑफ chromosomes of male pronucleus and chromosomes of female pronucleus. मेल प्रोन्यूक्लियस मधले 23 गुणसूत्र आणि फिमेल प्रोन्यूक्लियस चे 23 गुणसूत्र यांचं त्या ठिकाणी मिक्सिंग होतं and ultimately there is a formation of 23 pairs of chromosomes or 46 chromosomes. This process of mixing of male pronucleus or chromosomes of male pronucleus and chromosomes of female pronucleus is called as the amphimixis. Okay, याला amphimixis असं म्हणतात. Now या ठिकाणी amphimixis झालं, याचा अर्थ गुणसूत्रांच्या एकूण 23 जोड्या एकत्र आल्या आणि पर्यायाने या ठिकाणी 46 गुणसूत्र तयार झाली. इथे मी दाखवतोय. समजा या ठिकाणी हे एकूण 46 गुणसूत्र तयार झाले. याला आपण सिन कॅरिऑन असं म्हणतो. सिन फ्युजन, कॅरिऑन न्यूक्लियस. अँड नाउ दी सेल फॉर्म. अर्थातच आता ही जी सेल तयार झाली, दिस सेल इज डिप्लॉइड सेल, दॅट इज नथिंग बट द झायगोट. अर्थात अशा प्रकारे या ठिकाणी झायगोट तयार झालं, ज्याच्यामध्ये 46 गुणसूत्र आहेत, डिप्लॉइड क्रोमोझोम्स आहेत आणि झायगोट तयार होणं. This is the end of process of fertilization. This is the end of process of fertilization. It means now the diploid zygote is formed and it is the first cell of development of embryo. इथपासून पुढे एम्ब्रियोची पुढची डेव्हलपमेंट सुरू होते. So let us talk about the significance of fertilization. Significance of fertilization. First, the process of fertilization, it stimulates the secondary oocyte to complete second maturation division and formation of true ovum. True egg means the fertilization or sperm penetration, it bring about the completion of oogenesis by the formation of ovum. Just I told you, that is sperm penetration brings about completion of second maturation division and formation of true ovum. Second, we can say that fertilization, it converts the vitelline membrane into fertilization membrane necessary to maintain monospermy. Monospermy means what? We have already studied. And it avoids the polyspermy. Next, during fertilization, the centriole is introduced into the ovum. Centriole is introduced into the ovum, which is already absent. In the ovum, and that centriole is necessary for the further process of cleavage, for the further process of division of zygote. अर्थात झायगोटचं आता पुढे डिव्हिजन होणं, याला आपण क्लिवेज म्हणतो. हे क्लिवेज घडून येण्यासाठी त्या ठिकाणी सेंट्रिओल असणं आवश्यक आहे, की जे सेंट्रिओल या ठिकाणी फर्टिलायझेशन मुळे स्पर्मच्या माध्यमातून ओव्हम मध्ये टाकलं जातं. आणखी आपण असं म्हणू शकतो that the fertilization, it helps to restore the diploid condition, in the new young one, because you know, during fertilization, haploid male gamete and haploid female gamete, they come together to form the diploid zygote. So it helps to maintain and restore the diploid condition in the new offspring, or we can say in next generation. Because of fertilization, the ovum become metabolically very active and it starts the further process of cleavage. So there are so many different significance of fertilization. So here today In this lecture, we have completed the study of entire process mechanism of fertilization, the different physical and chemical events taking place during fertilization, and before to that the pre-fertilization events, and here we have discussed about the significance of fertilization. So here ends this topic. The next topic is cleavage, that we shall study definitely in our next lecture. So thank you and take great care of yourself.